Welcome back, everyone. So we're going to begin panel discussion number 2, which will be headed by Bjoern Peters from the La Jolla Institute for Immunology.
So somebody needs to stop screen sharing so that I can start sharing. Perfect. And this should be my screen. And I'm going to go into presentation mode. Okay. So as Jamie just said, this second panel is about how database providers can make COVID data fair. And arguably, database providers are at the front line of making data fair in general by making it accessible, findable, comparable, interoperable, and all these things. So the issue with COVID, obviously, is that it kind of changed the urgency of getting data into databases and making it accessible rapidly to their user communities. And that can be in conflict with goals such as fair, because some of the processes that have been developed to make data fair might not be feasible to implement in the times of a pandemic. So we will discuss in this session the challenges that different databases had, the approaches to engage user communities, and the lessons learned, ideally, to prepare for the next pandemic, because if there's one thing that's sure, that there is going to be one. So we have 5 very different databases that are represented here. I'm going to talk about the immune epitope database, the IEDB. Felix is going to talk about the iReceptor repository. Patrick Dunn is going to talk about IMMPORT. Erica Ollmann Saphire is going to talk about CoVIC-DB. And Darren Natale is going to talk about UniProt Pro. So these are very, very different scales, user communities, types of data, but they're all dealing in some way with COVID data. So each of us is going to give us an introductory presentation that should be like 4 minutes to say, what is your database about? How are you implementing fair data standards in general? And are you and how? And what were the challenges as lessons learned when you had to deal with COVID-19 data? So at the end, after this, so this should be 20 minutes or so, we're going to go for a panel discussion. So please put your questions into the Q&A. Go ahead as we talk. Also the panelists, I encourage you to put things in there as you come across issues. I like to prioritize questions from the audience. And also, as you all know, if you organize a session like this and you have 5 people presenting short, that's a challenge. So we're going to do a technical experiment, and I have all the slides on my laptop, and we are going to go through an auto advance mode. So I apologize in advance if speakers might be weirdly cut off by slides advancing, even though they didn't think about it. And so if speakers, I can speed up advancing, but I won't slow down and won't go back, so that we make sure that we are on time in the end. So again, like, this is a bit of an experiment, and I hope it's going to go okay. So with that being said, I'm going to start my own presentation about the immune epitope database and analysis resource. So everything I'm going to show you here is available at www.iedb.org. The key part is the database, which contains experimentally derived epitope information. The scope of the IEDB includes infectious diseases, pathogens, COVID, and other things, autoimmune diseases, allergy, transplantation, and cancer, although that's only a pilot at this point. We have B-cell and T-cell epitopes and MHC ligands, which are peptides that are binding or processed and presented by MHC molecules. And we have epitope-specific BCRs and TCRs. That is the link to the AIRR community. And I have to stress, we don't capture full repertoires, but only receptors that have known epitope specificity. Our data comes from manual curation of the literature. Journal articles are 2/3 of the data, and direct submissions, mostly by NIAID-funded contractors, about 1/3 of the data. The current content is over 1 million structures, so that is like epitopes, ligands, and also including things that were tested and negative in over 2.5 million assays from over 20,000 references. And then this data is used to generate tools that are made available in the analysis resource that can be used for predicting epitopes and analyzing them automating common workflow steps. So how do we make IEDB data fair? So the data we get is in journal articles, results, methods, tables, figures, supplemental data, a lot of supplemental data these days, and it's essentially free form. And so our goal is to translate that into a structured format in the IEDB to make it comparable no matter where the data came from, to have it in one format, and then identifying what are the epitopes, antigens, receptors, etc., that were identified. So this essentially means breaking data down in key value pairs, and then the values should ideally be coming from community standards, preferably OBO ontologies, and that are then used to connect the data in the IEDB to other resources. So what did we do with COVID? So when this came out, we started, as we have for other pathogens, to do a quick analysis of the data that is in the IEDB, and there wasn't any for COVID-19, but there was data on SARS-CoV-1. So we identified known epitopes, looked for homology, mapped them to SARS-CoV-2, predicted epitopes for CD4 and CD8, and assessed similarity coronaviruses. That was then made available online and on the IEDB webpage and bioarchive, and led to a series of publications where we then followed up on this kind of data, including experimental studies in Alice Zetter's lab. What challenges did we encounter in this whole process? So as I said, we are relying in the IEDB on community standards for knowledge representation, but the problem is that these were obviously not anticipating SARS-CoV-2. So NCBI taxonomy, which is really foundational to much of the work most of us are doing, considered SARS-CoV-2 a strain of SARS at the start, which caused all kinds of problems. UniProt had a great patch release with new proteins and annotations, but we were not up for dealing with such a patch release. We were having the regular disease timeframe, then there were disease state and stages. Everybody talks about mild, moderate, and severe disease, and it's completely unclear what that means. Different people have different standards for that, and actually use things like hospitalization, level of care, need for oxygen, and those kinds of things that we haven't been capturing, but that really seem critical. There's COVID-specific assays, such as ACE receptor blocking. Pseudovirus neutralization should probably be distinguished from true neutralization. And then there's other issues like that the information made available actually largely comes from preprints. So preprints, there was a flood of it. We all know this. We are only curating published peer-reviewed papers, so that caused issues. And then there's specialized databases emerging, such as ImmuneCode from Adaptive and Microsoft, and that required us to essentially develop new processes to connect to that kind of data that is becoming available. So that was my attempt to do my auto-advancing in slides, and with that, I would ask the next presenter to please turn their video on, and I'll turn myself on mute.
So, so great. Thanks a lot, Bjoern. I'm Felix Breden, and I'm going to talk about the AIR Data Commons and how iReceptor Gateway integrates the AIR Data Commons. One of the main goals of the AIR community is to provide, to curate and analyze all the AIR-seq data, and this is done through what we call the AIR Data Commons, and then iReceptor presents one implementation of this. This is all done through the iReceptor team in Canada, and then also the large iReceptor Plus consortium that's funded by the Canadian government and an EU Horizon 2020 grant. So, are we forwarding? So where we come in being fair really is through the AIR community working groups. The working groups do a lot of the work. I'm going to mention 3 of them that are most relevant to the AIR Data Commons. And this is the Minimal Standards Group, which presented a set of metadata standards that describe data that are either AIR-seq data that are either being published or put into a MyAIR compliant repository. The Data Representation Working Group, which does file formats for their rearrangement data. And then the Common Repository Working Group, which has developed the AIR Data Commons API, which then allows integration of these data. So in this slide, we're talking about the AIR Data Commons, which we envision as a set of MyAIR compliant AIR-seq repositories according to the FAIR principles. And then the one implementation or one way to query these are the iReceptor scientific gateway, which allows for querying these through either metadata or through sequence. So for me, it's interesting that you have the, on one hand, you have the AIR Data Commons, which is one set of repositories, but then you could have multiple implementations that allow querying of this. The good news is that we're growing the AIR Data Commons. We have up to 4 billion sequences once VDJ Server, one of the larger repositories in the AIR Data Commons, gets the COVID-19 data from the adaptive / Microsoft project that Jonathan talked about yesterday. And then we had an exponential growth in the COVID-19 specific AIR Data Commons. We have up to 500,000 COVID-19 sequence annotations, again, including the data from the adaptive biotechnologies. This includes up to 17 studies. And you can see a huge increase in the user downloads once we started getting the COVID-19 data going, and the iReceptor users per month. So in summary, the lessons learned, I think the most important thing is how willing researchers have been to share their data even before publication. They're reaching out to the iReceptor group to share the data. They provide original annotations. That gets back to what Charlotte Dean was talking about, rather than having to re-annotate all the data. The challenges that the AIR community, I think, sees here is that broader adaptation of the MyAIR standards, adoption of these distributed repositories model, continuing the sharing culture, and then the sustainability of these resources beyond the, into the future. So to share data or to obtain an iReceptor Gateway account, you can write to support at iReceptor.org. And that seemed to go rather quickly, but
it is so weird.
It's very weird.
It's great. And
Okay.
Apologies in advance again for this weird format. Also,
Yeah.
something seems to mess with the advancement. I think at the moment I'm using any kind of other controls, the site says stop. So again, this is an experiment. Patrick is up next, so I'm hoping this goes better.
Thank you for the invitation to speak on behalf of the IMMPORT team. IMMPORT is an NIAID date-funded repository. We collect, curate, and share biomedical research data with standardized content and format. We focus on immunological and clinical data sets with links to public repositories for genomics and transcriptomics. We hope to help in bioinformaticians find and use the data more effectively, and to introduce other investigators into open source data management tools such as Python and R tools. So I'm showing the data capture site on the left in orange, and then the IMMPORT shared data site where you can query and download the data in blue on the right. This is an interactive data model that we have on the website, and it has many elements brought from multiple sources. On the right-hand side, you'll see a listing of most of the reference sources for standardizing the terms that we have. We try to use ontologies whenever possible, OBO sources, of course, but we also use NCI terms, thesaurus, we use NCBI taxonomy, mesh terms as needed. We have multiple tools to make it possible to find and access the data. So we have a COVID banner, much like multiple other sites do, to indicate and provide links to the COVID data sets in IMMPORT that are available and other data sets that might be relevant for you. We support a free text search using Elasticsearch Index of descriptive information that's provided during the uploads. And we also have faceted search using standardized terms, so ontologies, mesh terms, that sort of thing. So by this combination of searching tools, we're trying to make it possible for people to find the relevant data sets and then move forward with that. there are multiple lessons learned. So, and some of these, we have valid goals, and sometimes they compete with each other, as noted by multiple other speakers. Patient privacy affects the willingness of an IRB to say when data can be shared, what aspect of the data can be shared, et cetera. It's necessary for the researchers to have time for data analysis, data generation, data interpretation. Then they have to prepare their manuscript and get it ready for publication, and then they have to do data sharing. And that's relatively the sequence of events. You can understand the relative priority of data sharing at any one time in their minds, and that's understandable. We've noticed, again, this COVID stuff has brought to our attention over and over again, that plan studies, data capture and sharing and curation is very different than analysis of clinical observations. Describing the when and why data is reported is very important. It may not come up in an EHR. That's not what they're necessarily designed to do. So putting that structure around it is very important. Noting key milestones in the data sets. So the observations, as Bjoern noted, hospital admissions, the types of interventions that were done, what are the outcomes for the patients? How is that all described? We need to have good data dictionaries to describe the names and the units for the results within studies, but also especially across studies, because if the units are not the same, you could be talking about an order or 3 orders of magnitude difference. And of course, there is always the issue of inconsistent and missing results for being reported for a data set. Especially true when people are trying fast and furiously to get the data shared. This is, there will be gaps. And so when we are curating the data, we come across these issues, and how do we deal with these inconsistent and missing results, and in conflict with trying to get the data out as quickly as possible and as reliably as possible. So FAIR for us includes making sure it's not only findable, but it's accurately findable, accessible. We have straightforward rules for access, and we need to get the data out as quickly as possible. Thank you very much, and we look forward to hearing about others' lessons.
Awesome, thank you, Patrick. Sorry, Erica, can you unmute yourself?
I just did.
Okay, awesome. So this is all you.
Great. I'm Bjoern's new colleague at La Jolla Institute for Immunology. I'm Erica Ollmann Saphire, and we are building a database to understand and now analyze and move forward antibody therapeutics against SARS-CoV-2. We were tasked to find antibodies that could save lives in impoverished people. And there are 2 main uses for which we would want these antibodies, either to treat an infection, to prevent it from progressing to severe disease, or to preventing infection. And that would be for anybody not vaccinated, not yet vaccinated, who can't be vaccinated, or in cases where vaccines simply didn't take, such as elderly and immunocompromised people, the people that we know are most vulnerable. In order to find what antibodies we think that we can mobilize to save lives, we need to understand which ones are the best. Now, of the 50 different competing companies in multiple academic labs, each forwarding these molecules, everyone says theirs is outstanding. So how do you know? How do you know which ones are best, what makes them best, and which in vitro assay best finds that? Well, the answer is that you have to negotiate with these people, get their therapeutics, and start to compare them side by side in multiple parallel kinds of analysis of every different way you might measure a function or a feature of an antibody and which ones correlate with protection. We're running all of the assays on the left in parallel, and all of the results will be gathered in the CoVIC-DB database, overseen, analyzed, and run by Bjoern Peters' lab. So there are a couple of key things here. How do you get the samples, and how you use the data from it? So key to how to get it is to protect the intellectual property and investment of all of these different, whether they're academic labs, startup biotechs, or multinational corporations that have invested quite a bit in making these therapies. Key to that is to anonymize all the samples. So everything in CoVIC-DB has a code name. Instead of the antibody's real name, they're just samples 1 to 200. And so that makes the database something that we can share immediately and publicly as we build it. So the body of information on the activities of antibodies at different sites and how they work together and what kinds of protection they elicit will be immediately and publicly available to all researchers around the world. The people that own the antibodies know which samples are theirs, but I don't, the PI, and the database doesn't either. So the goals are these. We have a translational goal: find and mobilize a cocktail to save lives in impoverished people. But while we have gathered all these competing molecules and generated this wealth of data, the academic goal is to understand what what about an antibody really confers protection against SARS-CoV-2, and to build that arsenal of information by which we could come up with a second generation cocktail and a third generation cocktail if the virus drifts, if there's mutagenic escape, or a novel coronavirus emerges yet again. So Bjoern, you can advance that slide. Give myself a couple of things. There we go. Okay, so how do you use this data now that you've got it? The example came from a very similar consortium we ran for Ebola virus when that outbreak began. We gathered 230 monoclonal antibodies from different previously competing groups, anonymized them, and built them into the database to compare them side by side. Because we were able to gather so much data on standardized platforms against all of the world's antibodies, we could build, for example, network correlation models to understand which antibody features track with other features, and more important, which things you could measure high throughput in vitro that actually forecast in vivo protection. So this is a logistic regression model. Large bars are strong predictors, either good in blue or bad in red. We could find better measures of neutralization that better forecast in vivo success than what we had previously analyzed. And we're able to find multiple features of the FC that strongly track with protection. And actually, had we sought antibodies based on their FC, we would have found just as important therapeutic candidates. So these are the experiments we're running parallel. They will be put into the database at the bottom, CoVIC-DB, and I'd be happy to answer any questions, and we're very welcome to feedback. If we're going to the effort to do this study, we want the data to last a while.
Thank you, Erica. And then I believe we have Darren. Darren, can you unmute yourself? Darren? I am not hearing Darren. Am I seeing Darren? I see Darren. Darren, you're unmuted, but I don't hear you. I am not sure what to do right now. Can I see Darren in the chat? Darren, do you have issues? I hear something. Hello?
Can you hear me?
Yes. Here we
Yeah,
go.
I had to remove my headset. Somehow it failed.
Okay, awesome. So, well, not awesome, but great that you got it to work. So I'll start you right now.
Okay. Hello, everyone. I'm Darren Natale. I'm from Georgetown University, based in Washington, D.C., and I'll be presenting on behalf of two resources. One, UniProt, is a database, while the other, PRO, or Protein Ontology, is an ontology. As you can probably surmise from the names, both are dedicated to proteins. UniProt's mission is to provide pretty much all there is to know about individual proteins, or at least include links to additional information. PRO's mission is to provide an ontological view of these, providing context, some increased specificity in the form of proteoforms, and increased generality in the form of taxon-neutral abstraction or protein families. As mentioned previously, databases and ontologies are already quite fair-focused, so I'll focus on our responses. And as I'll demonstrate, both UniProt and PRO have responded to the need for information on COVID-19 in similar manners. So the first step taken in each case was to collect any available information to create the entries describing the relevant proteins. As was done for IEDB, most information comes from the literature, but we also make use of prediction. There's no need to read the fine print here. I just wanted to show that regardless of our favorite editable format, we each convert the information to some widely used standard data exchange format, either XML or RDF XML, hence contributing to making the information fair. Both UniProt and PRO are released on schedules that span months. But that means that the collected data, had we stayed on schedule, would not make it to the scientific community in a timely manner. Accordingly, both UniProt and PRO created special data sets that were made available mid-release cycle. Indeed, and as is indicated directly in the UniProt case, these are allowed to evolve independently of the rest of the resource. In both cases, SARS coronavirus 2 proteins and affiliated information were made available both as a special search and as an independent download. UniProt actually took it a step further by creating a completely independent web portal to house the information. That'll come up on the next slide. And here you see it includes a download link highlighted in red on the left-hand side, and the ability to specifically grab the proteins of interest from the organism of interest. And that includes the host and the prototype SARS virus, and links to other resources. The links are the resources shown at the top right. But with the backend needs up and running, the new resources needed to be announced. And both UniProt and Pro opted to do so on their respective homepages, using colors that contrast with the overall scheme and placed in positions of importance, both of which were designed to bring attention to the newly available information. In the case of UniProt, other forms of dissemination were used as well, including YouTube, Facebook, Blogspot, and Twitter. And of course, both resources sent messages the good old-fashioned way, no, not snail mail, but email, including to potential collaborators. And the last slide just summarizes the actions taken by each of the resources I described. One thing I didn't previously mention is the license, which is in each case CC by 4.0. The main challenge that we face is due to the forking approach that we took. That is, we now have to maintain multiple sets of information, or in the case of UniProt, multiple non-equivalent web resources. I also provide in the middle there the links to all of the discussed pages. And I just wanted to point out lastly that UniProt is provided by a consortium consisting of the European Bioinformatics Institute, the Swiss Institute of Bioinformatics, and the Protein Information Resource, while Pro is provided by the aforementioned PIR, the Jackson Labs, University of Buffalo's Center for Excellence, and Reactome.
Perfect. Thanks, everyone. I asked the panelists to please start their video, including myself. And that went better than I thought. So thanks everyone for being willing to participate in that kind of experiment on how to do this. And we stayed on time. So the goal was to have 20 minutes for discussion. We have 23, so perfect. I have a few prepared questions that we're going to ask, and then I'm hoping, so we don't have open questions yet. So if somebody has questions, please put them in the Q&A. So looking at the prepared questions that we had, so after you have seen what everybody has been presented, what do you think is the most urgent need that we should be making better to, for the next pandemic, be ready and not have some of these issues? How would you prepare your own resource differently, and or what kind of resources would you build before the pandemic, and or what would you expect and hope that other people would be doing differently? I mean, other people, I mean, other resources. What would you wish would have been there? And to just start from myself, for us, definitely, it would have been nice if the NCBI taxonomy would have been rapidly updated. It took months, and it ties to everything. Every gene, every protein gets annotated with NCBI taxonomy IDs, and there was a consistent confusion of what is SARS-CoV-2 versus SARS-1 and these kinds of things. They're an awesome team, and they're badly underfunded. It would be, and there's so many people building on them. That's one of the things that, in my mind, it would have been great for us to have in place. So, without putting anyone on the spot, Felix, what do you think?
Well, I mean, I think at least from the viewpoint of the Air Data Commons, I mean, things worked pretty well, really. It showed a change in, as I mentioned, a change really that people were willing to work with us more closely and providing their data in a best, you know, most easily usable way. So I think really it's just a continuation of this willingness and kind of getting that more into the culture that, yeah, this is the way to do it. So I can't really put my finger on it. You know, it's just kind of moving ahead with the way we've been going now. But not, you know, probably what so many things will happen is that the momentum will go away, and then we'll get caught, you know, 2 years from now with a new pandemic. So I guess maybe the main important thing in my mind is to not let the momentum and we forget all the lessons that we've gotten. So, yeah.
Great. Patrick, how about you? I'm kind of going in the order of the presentations, how they were. So, Patrick here, yeah.
So building on what Bjoern and Felix said, so folks who are used to doing data depositions do it much more efficiently. Our first data set, I emailed somebody in China and they sent me a spreadsheet. And then they sent me a second version of the spreadsheet with the data dictionary. And I parsed that and put it in. But we work with the IPSI team, Bjorn's team, they've got a stellar team of data depositors. And so it's going to be much easier for their data sets to go into IMMPORT than anything else. And therefore a lot faster for us to share it. And it'll be high quality. So having this concept that data sharing is incredibly important for everyone, and having that a part of our psyche, our new psyche, I think that will make us more ready for the next pandemic.
Erica, you're obviously kind of in a different boat to some extent because your resource is COVID specific. But obviously, I mean, there's a backstory. Maybe you can elaborate.
The backstory on Ebola.
Well, yeah. I mean, how did CoVIC-DB come about? That was,
Well, CoVIC-DB came about because we had been running a consortium to find antibody therapeutics for Ebola and Lassa. The idea that, you know, for emerging infectious disease and global health issues, there's just not enough dollars. No one's going to make any money. And so if we're going to make an advance, we need to pull together and do a better job. And that worked very well. The Gates Foundation rather liked what we had built. And so they tasked us to make sure that of the antibody therapeutics being mobilized for industrialized wealthy nations, to make sure we could find molecules that were affordable and potent enough that we could save lives in low and middle income countries as well. And so we set up a second market parallel effort to try to work with academic labs to multinational corporations in order to make sure that those molecules could be mobilized as a charitable effort as well. And I want to echo what Patrick said. I think that the biggest issue is that we need the expectation in the human psyche of how data sharing benefits them. There is an expectation to want to hold your molecules and hold your data. And it takes a lot of conversations to have them realize that we are not competing with their effort. In fact, we are providing them lots of complementary data that they can have and they can use for their IND filing at no cost to them. Everything is protected, everything is blinded. And there is an opportunity for a dual market, that there will be people that will mobilize their molecule, and maybe it's their second choice molecule, and maybe it's their first choice molecule, and maybe it's a combination that's even better than anything anybody had discovered in their own wheelhouse. And we would only find that combination if we put company's molecule 1 with another company's molecule 2, and that can only happen if they're all in the same place. And so to get it off the ground, it's been months of multiple conversations explaining what we were doing, the value added for them, the value for the scientific community. And at the end of the day, they're all scientists and they're doing this because they want to make a difference for human health. But getting all of the legal ducks in the row is critical to get all the companies' lawyers on board and allow the sample sharing and the data sharing. Having that expectation from the beginning would save us months.
Thanks, Erica. Darren, how about you?
So we're kind of fortunate in the sense that we deal with proteins and not with any clinical data. So the information that we get is made available very early on. So one of the first things, you know, aside from the publications about the spread of the disease, one of the first things that came out was the genome and the proteins. And that's what we get and that's what we deal with. And we have a very large team of curators who read papers. So a lot of the problems that might come up with other types of resources we just don't experience. One of the things that I think is important to mention is not a problem, actually was something good that happened. I noticed that quite a number of journals, this has nothing to do with my database or anything like that, but quite a number of journals made available free, and even newspapers and whatnot, made available freely to all the papers about COVID-19 and SARS coronavirus 2. So I don't know if that came up earlier at all, but that was pretty good, and that's something that we got right in terms of what to deal with in the future. The information was just made available, so that was done right. And like I said, for us, we had the information available to us early on and acted on it rather quickly. I was somewhat surprised at how fast we got up a whole new, basically a whole new website. So yeah, I think actually it went very well this time around and can serve as a model for the future. I'm not quite sure what else could be done to improve it at this point, at least from our perspective.
Just any chance, Darren, to have a regular release cycle that is weekly?
Impossible to do.
Okay. Well, I can ask for the impossible. Okay. I understand. Obviously, you have like a massive amount, a network of knowledge that you have to rebuild, I guess, from scratch every time as you release.
That's correct.
Okay. Okay, so the second question that we had circulated previously was, so specifically because this is supposed to be about FAIR. So data standards work that like define, say, the vocabulary to represent certain things work best when they are built based on consensus and are centralized. And that always takes time, versus pandemics require rapid responses. So how should these be balanced? Again, I actually should have kept the NCBI answer for now. I think there's, for NCBI taxonomy, it would have been nice if they had just gotten something out earlier, even if it's crappy, and be fine with redoing it. But there's a lot of other resources, and yeah, I mean, how is there an issue with, I mean, okay, I'm going to ask Felix first again. So obviously for AIRR, there was a lot of consensus work being done to define like the minimal data standards or so. I don't know if there was any need to adopt them or so for COVID. Has there been any issues with this? Do you see any, what are your thoughts?
Yeah, again, I don't think we had to, we had to respond in any way there. I was thinking, one of the things, and I didn't explain it that in detail, with the AIR Data Commons, it's a distributed set of repositories is the model, at least. Right now we have two major repositories, at the iReceptor Public Archive and VDJ Server. But ultimately, we want to develop a federated set of repositories where people, wouldn't have to go to this central repository, but could put up according to the, if you accept the AIR Data Commons API, then you can put your data up and it can be immediately queryable through gateways. So, that's one thing I would push, is that you don't have to put everything has to go through this central repository.
Right, I guess let me rephrase that then. So how would you, for example, represent these are antibodies binding to the receptor binding domain? And then there's different definitions of that, and that's one of the kind of the things that you want to standardize so that your data and what Erica has in her database are actually comparable.
Well, I mean, we go back to the MyAir standards. They would have to come up with, we right now don't curate binding data, particularly in the database model. So we would, I'm not sure if I'm answering your question, but that would take a whole, that's not one thing we do. Right now we just curate,
Well, I was trying to find things that were specific for, or say you have repertoires from people with mild versus severe disease. How do you distinguish that? Or is there no attempt being made at all?
Yeah, no, I mean, that can be into the metadata but it's not in, we don't have a column that, right now, describes that. So yeah, I don't know how well we just describe the medical phenotype like that. Yeah.
Okay, Patrick, how about you?
So one way we deal with this need to get stuff shared as quickly as possible, but it's also standardized. We have a notion of a reported value and then a preferred value. That's not unusual within the clinical data realm. And so we put things into, or we allow people to use free text in many cases, and we recommend standardized terms in the reported value. And if we can map it to the preferred term automatically, excellent. Otherwise, we go back as a standard exercise and look through things in reported that we don't have mapped to preferred and try to do that mapping. So we try to improve the annotation over time so that we're not holding up stuff from being shared, because people could do that themselves. And our goal is to make it better over time so that they don't have to do that aspect. But people are right now willing to do whatever it takes to get the data and to analyze the data. We fully agree with that, and we are sending out the data as we get it, essentially, with the goal of making it better going forward.
Thanks, Patrick. Erica, how about you?
Well, we're pretty pragmatic. We don't really care too much what things are called. They just have to be called something and something intuitive that relates to what's being discussed in the literature. So we are going with antibody contributors' own names for RBD A and B and so on. And then the challenge has been getting data into standardized formats, getting the submission templates all worked out so that data can come in smoothly and easily and cleanly with minimal human intervention. And important to that has been the IEDB people in figuring out what the right template is and trying to get the bandwidth of the people running experiments at different places of the world for this consortium to figure out how that template should be, how the data is going to be presented so that it can come in smoothly, cleanly, and accurately.
Thank you. Darren, how about you?
So we have a pretty strong network of collaborators in UniProt and Pro. Chief among these for UniProt would be the nucleotide resources like GenBank and DDBJ. For Pro, we have a collaboration team, not just within Pro itself, but also within the OBO Foundry, that Pro is part of. And so in all of these cases, we have rather long-standing, streamlined processes. And part of those processes includes, communication and collaboration. One of the things that really ramped up in the beginning of this pandemic was the amount of collaboration and discussion between collaborators on how we can address everything, how we can make things available to the public. And as I said, the principal thing that we did was to fork the data so that anything that we had can be made available pretty much immediately. That's how we tried to make the information that we have fair, was to just get it out there fast. The other thing that we did was we do make extensive use of things like synonyms. What are things called in the literature? Well, they're not always called the same thing, and we have to capture all of that. So if your favorite paper is talking about Wuhan coronavirus, well, we have to make sure that we have that indicated somewhere, or what was it? The Chinese virus?
China virus, yeah, I was going to say.
China virus.
Or a virologist in chief thinks it's the China virus.
Yes, exactly.
Thanks, Darren. So I guess I should be pointing out, I'm comparing about, I mentioned the NCBI taxonomy a few times, but there was a lot of other resources. And we are making extensive use of actually then term requests to other related ontologies or so to get the data in. And it takes a lot of time. And these are also chronically underfunded. These are volunteer efforts. So it can easily take half a year. So these are not the time scales to get something in for a pandemic response. So I think what Patrick was saying and Erica was saying, that ultimately in the short term, we have to put in something that is not interoperable and clearly defined. And because there's no point in saying it's going to take us 2 years to have that standard and not share the data in the meantime, even if it's not perfect. Okay, so one of the next questions I had here And again, I would encourage anyone who has something that I didn't prepare to put it in there. But one of the things that came up are like privacy concerns. And as we heard actually in some of the previous day talks, that actually antibody repertoire data itself can be used to infer things like HLA type of a person. So which by definition then means, if you look at genomic data sharing, that makes a personally identifiable information potentially in genetic typing. So these concerns about privacy, also Patrick mentioned that, it can conflict with open data sharing. And so what are your personal views on it, starting with Felix?
Well, so the AIR Data Commons, as you know, has made a pretty strong statement that they're working with completely open data that's already in the public domain. I mean, I think it's a huge question, of course, and I think we do need to, like the iReceptor Plus project is going to try to develop levels of security on the repositories so that we can, you know, open up to more data sets that are more protected. You mentioned, we only have a few minutes left, you know, and a lot of people have mentioned how their communities have been so open with sharing because of COVID. And I guess it's because it's such a, you know, an intense threat and everything, but you do kind of wonder why. I mean, most of this biomedical data is there to be used for, you know, disease diagnostics and therapeutics. And you kind of wonder why, you know, people have, it's taken this to make people share. I just wanted to throw that out there. You know, it's almost not understandable why it's taken this to, you know, open the sharing. But anyway.
Thanks, Felix. Patrick, I know this is near and dear to your heart.
So as I noted, we sort of allow others to take care of the most severe privacy concerns because we don't take that data. So we don't take anything HIPAA or personal identify, dates, names, etc., addresses. None of that comes to import. We're getting data sets that are intentionally not designed to be identified. So the genomic issues, the transcriptomic issues, they're all going to repositories that have those things in place. So it may take you longer to get that data from, because you have to go through the data access committee, etc. The data to import, when it's shared, if you have an email address, you can get the data. That's all we actually ask for. So we can report who's getting the data at an aggregate level and how many people, that sort of, and which data they're getting. So our clinical data sets historically have been fairly thin. Now that we're getting more clinical data sets, we have to go back and review, is this potentially identifying? And that's a work in progress because we're still getting the data sets. Right now, they are still fairly thin. And we have to make sure people don't put dates in for hospital admission. We have to make sure that the date of intervention is not present. The name of the drug, yes, but who did it? Who's the technician? Who's the physician? Who's the nurse? None of that should appear, but we have to look to make sure that it doesn't appear. And so we actually get the advantage of working with Bjoern's team and others at HIPSI because they're doing a lot of that data scrubbing for us. We review what they send us. So we have 3 or 4 passes of making sure that we are not violating privacy and protecting study subjects' privacy rights because it's incredibly important. And that will affect what data is shared with whom and when. Those are all valid. And those sorts of policy decisions have to go back to the IRBs and NIH and the governments, international, to decide how they want to do this for the next pandemic. Because Ebola equivalent will come to us in some way. Zika, dengue, Zika, all of those are on their way. The next COVID is on its way. So how do we set ourselves up to answer these questions ahead of time so that the IRBs have those sets of questions to ask and decide, not just at an institutional level, not just at a state level, national and international.
Thanks, Patrick. And I think that meshes well with what Lindsay put in the Q&A, namely that the AIRR Common Repository Working Group has actually been trying to develop standard consent forms to make sure that this happens. It's also, I mean, looking at our own procedures, we are asking IRBs who are medical professionals to deal with highly complex information technology issues about what is identifiable or not. it would be so much better if there was a globally reviewed non-problematic consent forms that can be used in advance of time, so that sharing really is unproblematic. So Erica, I know, I mean, the data stored in your case is probably not as privacy related, but I mean, you have like the one Ebola case that came to the US and everybody knows who the antibodies are from. Or is there anything else you run into with along these lines?
Well, the issue is not so much patient privacy, but corporate intellectual property with our samples. That these companies have put a lot of investment into these molecules, and they're taking a risk of putting them in the hands of academics to look at them. And so we've had to put in multiple layers of protection to allow them to participate. And so one layer of protection is the antibody just gets a code name, CB1, CB845. The antibody owner knows which are theirs. I do not. So I will never write a paper saying such and such is better than so and such. The academic paper will be written, or the antibodies against this epitope have the following FC features in this range of activity. Another layer of protection would be if an antibody owner felt that their antibody could be identified based on the data characteristics. So one company had a particular FC modification that would have inspired very special functions, and it could have been found in that database. They said, well, just send us a bunch, bury it in there, give a different Fab on the same FC, give me your clinical Fab on a wild type FC, send me 20 antibodies, we don't care, it's more data for the database, more academic information. But the antibody owner gets back all of that information, and then the NIH program officers are using our independent standardization to choose which things go into their clinical trials. They have access to the confidential information, so they have the independent verification they need. And the Gates program officers also under confidentiality agreement can see what they are. So there's multiple layers of protection.
Thanks, Erica. And very quickly, Darren, I think you already said that you don't deal with it because you deal with proteins who are famously public, but please.
Yeah, we don't deal with any clinical data at all, so there's no scrubbing necessary. It's scrubbed well before we get it. The only thing we ever have to worry about is making an assertion that such and such a protein has some involvement in such and such disease. And then we have disclaimers saying, don't take our medical advice. That's the closest we come to dealing with clinical information.
Okay. Thanks, Darren. Okay, so we are 2 minutes over. Thanks everyone for your participation in this and the weird experiments of auto advancing slides. And I'm very happy with how this went. Thanks everyone again. And I think we have a 10-minute break now.